Hello, dear friends, and today we'll speak about amazing medicine, about monoclonal antibodies. Creation of monoclonal antibodies led us to revolutionary achievements in diagnostic and treatment of serious diseases. Among them are tumor, systematic autoimmune, and cardiovascular diseases, some types of infection, and others. Besides that, monoclonal antibodies are widely used in diagnostic. What is so amazing about monoclonal antibodies? Some historic background. The problem was that scientists has been looking for a solution to get antibody for a specific antigen for a long time. It all started with attempts to immunize animals. We all remember the great Emil Behring, who worked with Paul Ehrlich and Kitasato Shibasubra and created antidiphtheral serum, which saved millions of people. Also, the antibodies which were created this way contained a lot of molecules with different specificity. In other words, antibodies directed against specific antigen used for immunization, and other antigen with donor animal got. This is why these antibodies are impossible to use for targeted therapy. For example, cytokine, cell signal molecules blocking which initiate inflammation. The solution was found in 1975 by scientists George Cochlear and Cesar Milstein, who produced monoclonal antibody hybridoma cells from B lymphocytes of a mouse. These scientists were awarded Nobel Prize in Physiology in Medicine in 1984. What happened? The scientists created somatic cells hybrids from mouse lymphocytes, antibody production cells, and tumor cells, a cells that divide and exist in test tube. It's very important because lymphocytes are not able to live outside the body. This hybrid inherited an ability for antibody synthesis from B lymphocytes and immortality and ability from uncontrolled growth from tumor cells. Selected hybridomas are cultivated to make a big number of monoclonal antibodies. The produced antibodies are so pure that they can be considered as homogeneous substance. It's important that monoclonal antibodies produced per one hybridoma culture and interact only with one antigenic determinant. It means monoclonal antibodies work accurately and connect only with definite receptor. In fact, George Cochlear and Cesar Milstein created a production line of targeted and standardized antibodies. The first monoclonal antibodies designed from a mouse cells had serious limitations for the purposes of treatment. Injection of these monoclonal antibodies led to immune reaction to heterogeneous mouse protein. However, in 1988, Greg Winter, one more Nobel laureate, developed special antibody humanization method. His idea was to replace some parts of animal protein with human protein. This way, the problem of immune response was solved and new antibodies got a name chimeric antibody. Chimeric monoclonal antibodies connect 65% human immunoglobulin in their structure. The next step is creation of humanized monoclonal antibody with the help of transgenic technologies. Humanized monoclonal antibodies contain 95% of human immunoglobulin. Nowadays, fully humanized monoclonal antibody already exists. To understand what monoclonal antibody we are dealing with is pretty easy. It's enough to read and name attentively. All name monoclonal antibody end with MAB, derived from monoclonal antibody. If an antibody was made from a mouse, then O is added and the ending is OMAB. Chimeric antibodies end with XIMAB, humanized antibodies end with ZUMAB, and fully humanized antibodies UMAB. So, for example, tocilizumab, which is used to cure rheumatology arthritis and also used to treat severe COVID-19 patients, this is humanized antibodies. Rituximab, used to cure B-cells lymphoma, chimeric antibodies, and so on. Let me briefly explain how monoclonal antibody works on the example of tocilizumab. The main indication of using this monoclonal antibody, rheumatoid arthritis, but now this medicine is given to patients with severe COVID-19. So, how does it work? The thing is that tocilizumab is interleukin-6 blocker, cytokines that is produce different types of cells in the human body, lymphocytes in particular. Interleukin-6 initiate immune response on pathological agents, including viruses, and damages. That's why inflammation reaction begins. So, by blocking interleukin-6, tocilizumab fully stops immune response and consequently inflammation process. Now it's clear why so-called cytokine storm when the immune system reaction is so strong that it can destroy its own tissues, this monoclonal antibody demonstrate significant results. This is just one example of targeted therapy, but over the last 15 years, more than 18 therapeutic monoclonal antibodies have been proved. Many of those have made a revolutionary breakthrough in diagnostic and treatment of oncohematology and other serious diseases. Dear friends, I have told you briefly about this amazing medicine. I'm sure that further development of monoclonal antibodies will save millions of lives. I hope you enjoyed my presentation because the pharmaceutical world is wonderful. Thank you very much and see you.